Hello, biology students. It is Friday, April 24th, and what I'm going to do is go through a screencast on meiosis and sexual reproduction. It kind of piggybacks on what we did on Monday, only I noticed that I didn't actually put the screencast up. That was my bad. So that's going to be your assignment today, is to go through meiosis and sexual reproduction, and I'm going to have it uploaded in Edpuzzle. So that way I know that you're going through it, okay? Because I know it's sometimes easy just to kind of space it out and say that you went through something, but I want to make sure that you guys are going through what we had up. Before I get started, I hope that you guys had fun with the Earth Day activity. I know it was kind of random, but I figured with us being a biology class, and sometimes it's a nice change up to get out and try something different. So I really enjoyed some of the memes and poems and songs that I got. It was quite interesting. So thank you guys for doing that, and I hope that you enjoyed that little time. But we are going to go back to what we did on Monday, and that's watching Life's Greatest Miracle. And so I hope that you weren't too uncomfortable with it. I stopped it before it got too detailed, and I know most of you guys should have had wellness by now, so hopefully it's not going to be too uncomfortable when your biology teacher is talking about sex and sexual reproduction, because we're going to focus on the biology of it. Before we get to that, I just want to remind you guys about some terminology. Also, I don't have a set handout for notes. So if you just want to take notes in your notebook, that is completely fine here, okay? So we're going to be just doing a quick review. Remember, chromosomes are long coiled up, and we'll see another picture in a few minutes, DNA strands. And different parts of the chromosome, or different segments of that DNA, of that chromosome, that code for different traits, are known as genes. And so you guys acquire the genes from your parents by inheriting their chromosomes. So each gene in an organism exists at a specific we call it a locus, okay? And so you've got one from dad, one from mom. They're in the same spot on the same chromosome number, and we'll talk about that in a few minutes, okay? But we inherit one set from mom and one set from dad. And we call these chromosomes that have the same genes in the same location, we call them homologous chromosomes. Hopefully you remember that we talked about the prefix homo, meaning same. So these are the same genes in the same spot on the same chromosomes, but within it, there can be variation within that gene, okay, and what it codes for. Really quickly, we talked about mitosis at the gosh, my time is going. I have no clue. When did we talk about it last? We talked about mitosis somewhere towards the beginning of the semester. And when we're looking at mitosis, we talked about how somatic body cells can undergo cell division. And it happens for two reasons. Primarily, one of them is growth, and the second one is repair. There are some organisms, such as bacteria, that also reproduce asexually, and they use mitosis as part of that. Now, there's some, and when I'm talking about this, it's a process similar in bacteria. We call it fission when they divide, but it's a process very similar to mitosis here. We're not going to focus on asexual reproduction. The thing with asexual reproduction is that a single parent produces identical offspring. And in the video that you watched, I hope that you noticed that sometimes that's not a good thing. If a disease comes, which we're going through right now, and everyone has the same exact genetic makeup for the antibodies that fight that disease, because we're all clones, we're all asexually reproduced individuals, and we have a trait that is not able to fight it off, then it could kill off an entire population. Sexual reproduction combines sets of genes from two parents, and that's a really positive thing when we're looking at natural selection and evolution and survival of the fittest. So really quickly, a review: chromosomes. Remember, they're just strands of DNA. They're wrapped around something called a histone, coiled up into tight strands, okay? So here's our DNA. If we were to take a chromosome and we were to kind of unwind it, okay, we have a chromosome, and we're going to be focusing on chromosomes that are going to have that are going to be undergoing meiosis. Let's say that, okay? But here's our chromosome, and if we were to unwind this, we could see that it forms the DNA double helix. And so this section might, right here where my cursor is, down to this section, might code for one particular trait. It might be one gene. Now, test question for you. How many chromosomes in a human body cell? Okay, hopefully you guys said 46. I don't have it written in the blank. 46 chromosomes in a human body cell. Not all organisms have that many. Some organisms have just 2 or 3 chromosomes. Some have quite a bit more than us. But in humans, we're talking of 46 chromosomes as the magic number. That's in our body cells. That's in our liver cells, our skin cells, etc. Now, there's also sperm cells and egg cells, which we call gametes. They have half the normal number of chromosomes. So half of 46 in this case would be 23 chromosomes for the sperm cells. Also, the egg cell also has 23 chromosomes, okay? So body cells have 46, but when we're looking at our gametes, the egg and the sperm, they each have half that number, 23. And there's a reason for that. Before we get into the reason, a cell that has half the number of normal chromosomes is called haploid. Okay, you can think about it as half haploid. Okay, if you have the correct number of chromosomes, you got copies from both mom and dad, then you're going to be known as diploid. Body cells with our 46 chromosomes are diploid. Gametes, egg cells and sperm, are haploid. Those are 2 vocabulary terms that you'll want to know. So the human haploid number is, if you said 23, that's awesome. The human diploid number is, if you said 46, you're rocking it out. Now that's for humans. So in the case of a fire-breathing yeti, okay, it might have 50 chromosomes. And if we were to be told that it has 50 chromosomes, the diploid number is 50, what would the haploid number be? Okay, if you said 25, that was awesome. The haploid number of a flying liger is 74. So what's the diploid number? It tells us that's the haploid number. So in order to get the diploid, we have to multiply this by 2. So you're looking at 148, I believe. So here's our gametes. We have our little sperm, and we have our big egg. These are not drawn to scale, but when our egg and our sperm come together, that's a process known as fertilization. Okay, this is fertilization. This arrow right here, when they combine, 2 haploid gametes combine to make a diploid cell, that's called fertilization. So we have a single N and a single N combining to make a 2N, a diploid cell. So each diploid cell, and you saw this word earlier, has a set of homologous chromosomes, homo meaning same. It's a pair of chromosomes that contain the same gene sequences. One's from mom and one's from dad. Okay, so we have our paternal and our maternal. This is before replication happens. Remember, the cell cycle, when we were talking about interphase, we have our G naught and then we have our S phase, we have our replication happening. It's still going to happen for our cells that are making our gametes. It's just going to be a little bit different here. So we're still going to replicate and get this nice pretty X. Now, all this is when we're It gets kind of confused. Okay, I always thought this was confusing. So this single one is a chromosome. This duplicated one, the one that's been replicated, is also a chromosome. Only when it's in an X shape, we refer to each side as a chromatid. Okay, and so that kind of gets a little bit confusing. It was something that always threw me for a loop, but just remember that if it looks like it's kind of connected in that X shape, it's still a chromosome. This is an unreplicated chromosome. This is after replication happens. So here we're going to use some color coding here. We have 2 sets of homologous chromosomes. We have set number 1, you can tell by the different size here. And we have set number 2, different size here. They're homologous because they have a gene in the same location on each. And oftentimes when we're representing genes, we use these letters. We call these alleles. And we're going to talk about dominant and recessive coming up. And it's just easier for us to write out a letter symbol than it is to actually write out the sequences for the DNA at that specific location. Now, in order for us to actually gain more knowledge about chromosomes, we prepare what's called a karyotype. A karyotype is just a display of chromosomes arranged in homologous pairs. And so we're going to look at this here. So here, remember, we have 23 sets from each parent, 22 normal ones, and then we also have a pair of sex chromosomes here. And I'll get to that. But notice you have 2 sets because this is the replicated form of the chromosome, one from mom, one from dad. If it wasn't replicated, it would just be just the one line. But this is what a normal karyotype looks like. Number 1 is the longest. It has the most amount of information in it. And it goes down to the smallest. So they're arranged by sizes. Notice that X is significantly larger than Y. If this was this right here is a boy, okay, it's a male. If it was XX, 2 bigger ones like this, then it would have been a female. If you ever get genetic testing done or know someone that has, this is what they will prepare. And it's useful in trying to identify chromosomal abnormalities. And so you can see if there's like an extra set. For instance, right here, notice that 21 has an extra set. There's 3 of them here. This is known as trisomy 21. It's also known as Down syndrome. And it's when there's an extra chromosome. It wasn't caught during meiosis and fertilization. And so that chromosome stays with that person. In fact, I had a daughter a few years ago who had trisomy 18, known as Edwards syndrome. And I actually have her karyotype to this day, where you can see the 3 sets of chromosome number 18. Okay, so here's another picture with Down syndrome. You can see chromosome 21 has an extra one there. So how are they prepared? You don't really need to know how they're prepared. If you take AP biology, you might learn how they're prepared. But basically, cells are treated with a drug to stimulate mitosis, and then they're grown in a culture for several days. So that way you have enough DNA that you can take an image of it. Once it's undergoing mitosis, because remember, most of the time it's actually an interphase. You want it to be in that mitosis phase, which doesn't happen for much of the cell cycle. But once they start going into mitosis, then they can be stopped in metaphase, and then they stain them. And that's what you saw here, is that they're kind of lined up in metaphase right now. So here, once the picture is taken of them, an image of the chromosomes is displayed, and it's used to arrange them in homologous pairs because they don't just always line up next to each other. And so they'll click and drag and find ones that are the same size with the same genes in the same location. So why is it important to study the karyotypes? Again, it's to screen for defective chromosomes, for abnormal numbers of chromosomes, but also just to see the gender. A lot of times people will want to know the gender of their offspring before an ultrasound is able to be performed. And so this is early diagnostic testing. Now let's go back to that fun word gametes. You remember our sperm and our egg, okay? The forming of gametes is called gametogenesis, and it happens by certain cells in your body. And I think I thought it was on this slide. It might be on the next one. If we're talking specifically forming sperm, like in the case of the males, in the testicles, you're looking at spermatogenesis. The formation of egg cells is called oogenesis. Remember, spermatogenesis is going to be happening throughout a male's lifetime up until they've peaked at their reproductive age, whereas the formation of eggs happens before a female is actually born. Germ cells are the cells that produce your sperm and your eggs, okay? Vocabulary-wise, I don't have this highlighted. If you know sperm, eggs, gametes, I think, and you know somatic cells are your body cells, I think that would be fine at this point. Now, these germ cells, though, that make the sperm and egg are the only ones that can undergo a different cell division process called meiosis. Now, meiosis has a couple of parts. The first part is a reduction division, where it reduces the number of chromosomes in half. Okay, so we're going to separate homologous chromosomes in a diploid cell. And so we started with 2N, and so now we're going to start reducing them down. Now, the cool thing with meiosis is this is where some of the variation starts to occur. When these homologous chromosomes line up, they get super, super close. And as you notice here, you should see that they start swapping DNA. Okay, if they get close enough, they can swap their DNA, and that's known as crossing over. And so now if you're looking at this paternal one down at the bottom, it is slightly different than dad. This maternal one is slightly different than mom. And it's a process called crossing over that allows this to happen. And that's one of the processes that leads to variation in organisms. Now, it used to be that we would memorize every single step and what it looked like for meiosis, but that is quite a bit. What you need to recognize is that it goes through PMAT twice. We have the reduction one, where it pulls the chromosomes apart, and then the chromatids, those little strands, single strands right here, are further split apart. You don't need to memorize what each of them looks like, but just know that mitosis only undergoes one reduction here, goes through PMAT one time, whereas meiosis goes through it 2 times. Now, because we're talking about the sexual life cycle process, we do have to remember it's involved in human reproduction. And there are a couple parts with that. There's meiosis, where we make our sperm and our egg. There's fertilization, where they come together. And once they come together, they're at that diploid number. Once we've had the haploid and a haploid come together, we're back up to our diploid number, and then we begin the regular process of mitosis. The zygote is the name of the cell when the sperm and the egg come together. And after it starts dividing, then they can start to differentiate, and the skin cells will start becoming skin cells, and the optical cells will start to become the optical cells. So meiosis, fertilization, mitosis, and then differentiation. And so here we have a juvenile. Okay, it's diploid 2N. Okay, meiosis in females and males occur. So we're down to a sperm, which is haploid, and an egg, which is haploid, only 1N. They come together, fertilization occurs. Now that it's been fertilized, this egg with the sperm, then mitosis can go ahead and occur. And mitosis, remember, growth and repair. And in this case, we're looking at growth from here on out. and cell differentiation. So this is a kind of lengthy slide here, but if you wanted to pause it and look at it a little bit more closely, hopefully that you see that this kind of jives with what I was saying. The synapsis means that connection where the homologous chromosomes become really close together, where that DNA is actually able to flow from one to another. We call that the synapsis. So how is it meiosis and mitosis different? The big part here is that meiosis, cells divide 2 times, mitosis once. Mitosis, body cells. Meiosis, it's just to make your sperm and your egg, your gametes. And most importantly, meiosis, what it provides in the big scheme of things is that it allows the normal species chromosome number by making those haploid gametes, those single end gametes. So that way, when they come together, the sperm and the egg come together, they're back up to the normal diploid number. This is the big part right here. The big takeaway too is it increases genetic variability by that process of crossing over and recombining genes in eggs and sperm. So there are a few terms here that I'd like you to know. Crossing over, recombination, independent assortment. Those are the 3 big ones right here. So crossing over was when they came together, when homologous chromosomes came together and exchanged genetic pieces. You don't need to know when it happens at this point, but that little connection where they meet up is called the synapsis. And here's another picture of crossing over occurring. They get super, super close together, and so now they can swap some of their DNA. Another factor that increases variation is recombination. It's when a new combination of genes are made on a chromosome during that crossing over. And because of that, you can get new combinations of inheritable traits that were not available before. It increases genetic variation. Okay. Independent assortment is each time a body cell goes through meiosis to make a gamete. It should be a germ cell here. All the chromosomes line up in a different way. And so it's this random assortment of chromosomes into each sperm and egg. And so it just offers more of a chance of variation. And here you can see here, again, don't stress about the names of the phases, but you can see they start off the same here, but it could divide out. So we have like all paternal, all maternal, or it could be like one of each. And so you increase combinations. Okay, and then this is another picture here, just showing that independent assortment. Now, the last thing I want to kind of end on here is that mutations are the original source of variation. So a mutation for a gene to have a new variety, there has to be a mutation to establish that variety for the trait. But once it's in a chromosome, once it's in the DNA, then meiosis and evolution can act on it, okay? But mutations are where all variation starts. Meiosis here, this formation of sperm and gametes, or sperm and eggs, is just a way to shuffle up these mutations once they are in the genome. And finally, I left these 2 slides in here. But a chromosomal abnormality is just when there's an incorrect number of chromosomes, which you saw with the Down syndrome. Depending on which chromosome it affects, oftentimes the body won't even be able to continue on with mitosis, and oftentimes those offspring are absorbed within the womb. And finally, we didn't talk about differentiation, and I'm not sure how much time we're going to spend in differentiation, but it's when the cells in the embryo develop a specialized form and function. So they start to have different jobs. So 4 and 5, I'm not as concerned with. I want you to focus more on the genetic variation parts. So let me know if you have any questions. Oh, here's just a picture of differentiation. Um, we are going to start our research project that I told you about last week. We will probably be ready to start it next week now that we understand a little bit about genetic variation. And I will get you that information out probably Monday or Wednesday. Take care.